All right, everybody. So welcome, everybody. This is Dapper Dino. I am here both on Ahmed's channel as well as on my channel, and I am having a slight technical difficulty with the image. So just give me a second. I'll have it fixed in no time. But we are debating whether or not evolution can result in novel features. And we are debating between Dr. Dan Cardinal, an evolutionary biologist and professor at Rutgers University. And on the other side, we have Finding Truth, Ahmed, who is an engineer from Egypt, I want to say.
Yeah.
Okay, good. So, and they are going to be debating this topic. Of course, Dr. Dan is going to be taking the affirmative, and he will be opposed by Ahmed, who is taking the negative. And so we're going to be having Dr. Dan go first. And Dr. Dan, why don't you go ahead, and if there's anything else you want to introduce yourself with, go for that. But then once you start actually making any arguments, I will be timing you for 12 minutes. And so both sides are going to have 12 minutes, and we're going to go to open discussion. And we're going to end off with about a half an hour or so of Q&A. So I look forward to hearing from both of you, and I hope not to have to moderate too much because I trust both of you. Don't let me down. Dan, go ahead.
Oh, thank you. I'm thrilled to be here. Thank you for hosting Dapper and Ahmed. Ahmed, really nice to be able to have a nice long conversation with you. I know we've crossed paths very briefly once or twice, but we've never had like a nice long conversation. So I think this is going to be a lot of fun. For those of you who don't know me, I'm Dan. I'm an evolutionary biologist. My channel is Creation Myths, and I talk about creation science. I lean very heavily on the genetic side of things. So most of what I do is about the common kind of genetics-based arguments that you hear coming out of creation science. And today we're kind of talking about that and kind of talking about related things. So our topic today is, can evolution or evolutionary processes lead to or result in novel features of some kind in an organism? And
Dan, can I stop you a second? I think we might be having a technical difficulty. Ahmed, are you attempting to stream to my channel as well?
Whoops, I need to kill it. Sorry, didn't do that.
That's okay. That's okay. Hopefully that will fix it. I'm going to refresh here on my end and see if that has fixed it. Because, yeah, YouTube was complaining at me. Okay, we should be back. Sorry about that. Okay, yeah, we should be back now. So, Dan, if you could just keep going on that.
Should
actually, you might want to start back over just a little bit, yeah.
I'll just go back a little bit. That's fine. Hi, I'm Dan. I'm an evolutionary biologist. My channel is Creation Myths. And normally what I do there is, my background is in genetics and evolutionary biology. So what I do there is focus on the more genetic-based arguments that come out of creation science. Today we're doing something kind of related to that. Our topic is, can evolutionary processes result in novelty, novel features, whatever that means. So I think the first thing we need to do is say, like, what do we mean when we say novel feature? And there's lots of ways you can talk about the features or traits of an organism. You can talk about like biochemical features. So like a protein catalyzing a specific reaction or having a specific binding site, right? Talk about genetic features. There's a particular gene that's present, or maybe there's a gene duplication that occurs, right? Physiological features. Do you do something, right, in terms of how the organism works kind of on a more macro scale? And of course, morphological, right? Can you just get like new parts of the body? So you have different levels of what you might call a feature in biological systems. But the question is, can evolutionary processes generate novel features? And for each of those different kind of ways of defining a feature, the answer is yes. And we know that the answer is yes because we have specific observed examples of them. So I'm not going to have any slides or anything up. I'm just going to kind of go through some of the examples here. So in terms of just like biochemical features, we have a protein called VPU. It's a tiny little protein. in HIV. And in one type of HIV in particular, VPU has evolved a completely new binding site and a completely new function that no VPUs have ever had before. This specific binding site that allows it to do a specific thing. So it's a completely new biochemical trait, and HIV has only existed for about 100 years. It crossed over from, I should say HIV-1 is what I'm talking about. HIV-1 has only existed for about 100 years. It crossed over from chimp SIV, simian immunodeficiency virus, in the early 20th century, 1920s and 1930s. And the SIV chimp version of the virus doesn't have this characteristic to it. It doesn't do this specific thing in the VPU protein. So that's a completely new biochemical trait that we know evolved within the last 100 years or so. We can look at specific genetic traits, and you can look, for example, in laboratory experiments like the Lenski long-term evolution experiment, the famous citrate metabolizing line of E. coli. The reason that that specific line of E. coli is capable of doing something called aerobic citrate metabolism, it'll use citrate as a food source in the presence of oxygen, is because of a gene duplication. You have a gene that you need to use citrate. We've got one copy over here, and it duplicated, and now you've got two copies, and the new copy was under a different regulatory scheme. Creationists often claim that you just break the regulation, you lost the regulation of it. It's not true. It's just the second copy is expressed under a different set of conditions compared to the first copy. So you have one that's expressed anaerobically without oxygen, and one copy that's expressed with oxygen. You get this new genetic trait, and it allowed that particular population of E. coli to survive, or I should say to grow to a much higher population density, because they are able to better exploit the resources in their environment. But even going up further in scale from that, we can look at something like lizards that give live birth. Now, this is something that's surprisingly common. We think of reptiles, both the scaly kind and the feathery kind, as laying eggs, right? That's kind of like the thing, or one of the things that defines for, kind of the average person. I want to get technical here, Dapper, because I know you know the specifics of it, but it's one of the kind of the things for when people think about reptiles, they think about laying eggs. Well, there's a lot of reptiles, the scaly kind, I should say, the non-avian reptiles, that actually give live birth. Now, some of those are basically developing the eggs internally rather than externally. But the really interesting thing is that we're actually seeing populations right now where either they previously laid eggs, and now some of the individuals are giving live birth through this kind of internal egg development process called oviviviparity. The most interesting kind of observation of that is a recent case where a single individual actually, within one litter, did a combination of laying eggs and giving live birth. So you kind of mix and match. And then in at least one population of lizards, we have now documented actual true placental development. So it's not just the eggs developing internally, but basically the same process. It's a completely different process where you have this interface between the blood vessels of the parent and the developing offspring. And that's something completely new for non-avian reptiles. It would be new for avian reptiles too, but we haven't seen it in them to my knowledge. And then we could just talk about like kind of new features of organisms, like morphological stuff. We've documented how feathers evolve, for example, and we could induce this in the lab. So you can get, for example, I think it's, I always confuse if it's alligators or crocodiles in the particular experiment, but it's one or the other. And you could actually tweak their genetics a little bit and cause them to develop tube feathers, which is kind of a rudimentary feather morphology. It's just changing the expression pattern of some of the genes that regulate how scales develop, but you can actually see that structure develop instead into a type of feather. And we've documented the regulatory pathway to go from a simple flat scale to kind of a tube feather to a more complex kind of modern feather morphology. And then the other really cool one that we've documented experimentally that I really love is obligate multicellularity in green algae that previously didn't have obligate multicellularity. These are algae that are unicellular. They can facultatively clump up, so you can have different individuals that would clump together into small colonies. But what we documented experimentally in the lab is if you impose a selective pressure like a predator, then that same type of green algae will become obligate multicellular. It will always be multicellular. And instead of assembling from different individuals coming together to kind of team up, instead what happens is the multicellular structures grow clonally, meaning you start with either one cell or a few cells, and they'll divide and they'll stick together and they get into a larger multicellular individual. So that's obviously one of the major evolutionary kind of traits in the history of life on Earth, is the evolution of multicellularity in a bunch of different lineages. We've now documented like that exact thing in the lab. And then there are a couple cool examples that are just kind of cool. So I want to mention them. One is a process called endosymbiosis, which is how a larger cell can engulf a smaller cell, and that smaller cell can become an organelle, which is just part of the larger cell. Instead of getting kind of digested, it kind of, the relationship stabilizes through a process called symbiogenesis. And we're now observing that not just in a completely new type of green algae, but also in a lot of animal lineages. So there's like a dozen animals right now that are at various stages of becoming photosynthetic, because this process of endosymbiosis is how structures like chloroplasts originated. There's only one type of chloroplast initially. Now we know of a second type of, we don't call it a chloroplast because it's a different lineage of organelles, but then that original chloroplast, there's processes called secondary and tertiary endosymbiosis, and that's happened dozens, if not hundreds of times. So you end up with all these different photosynthetic lineages, and now we're seeing a number of different animals undergoing that process now that we know what to look for. And then the final thing I'll mention is just a super cool experiment from just a few years ago. We have something called a plasmid, which is just a little circular piece of DNA that hangs out in bacterial cells. And normally it transmits through, it would be like a pilus, like a little tube that connects two cells is the main way that plasmids are going to transmit. Well, we've documented that, we've now documented this form of extracellular transmission of plasmids where they kind of blob off in these little lipid bubbles to move from cell to cell. And that's basically a plasmid evolving into a virus before our eyes. This was an experiment. It was actually in archaeal cells rather than bacteria. And it's so it was an archaeal plasmid. And this transmission back and forth via these virus-like structures. And that had never been observed before, certainly not in the plasmids that were being used in that experiment. So they evolved in the experiment a novel transmission method from cell to cell. So there's a lot of different ways we could talk about biological features, but the common thing is no matter what kind of level you're looking at, you can look at very small scale biochemical, you can look at very large scale morphological traits. No matter what you're looking at, we have documented examples of new features evolving like in real time. We don't have to look historically, we can look in real time and largely at experimental instances in the lab to see the evolution of new features. So can new features evolve? Yes, they can. Thank you.
All right, with just about 2 minutes to spare, that is Dr. Dan. So, Ahmed, if you want to, you can take just a little bit to introduce yourself, and I won't hit start if you do. Otherwise, I'm going to start going once you start giving an argument or when you show your slideshow.
Okay, so I'll do two things. So I will defer responding to the examples that Dan has said after I do my presentation in the back and row. And I'll just do the presentation, and then maybe we'll start by responding to what he has said, and he can respond to what I've said, etc. So let me see if I can share the presentation first. It will have my introduction in the beginning. You can... Okay.
I won't start during your introduction.
All right. It's going to be short anyway. So, yeah. Share screen. Window. Do we have it now on the screen? Okay, great. So, yeah, so I am an electronics kind of communications engineer. I did my master's courses focused also on computer and control. And I didn't do my thesis, so I don't have Masters of Engineering, but I did Masters in Business Administration, focused on international business, and 4 years of Islamic studies. I have 30 years of experience with information systems design and implementation and implementing complex IT systems. 20 years of which I'm an executive. I speak 4 languages with different proficiency, travel to almost every continent in the world except Australia. And my worldview is obviously I believe in God and I'm a Muslim, and I have the Finding Truth channel, which has 30,000 subscribers. So this is the introduction. You can tick the button. So, as an engineer, I believe in design. Engineers have the privilege of working with designing things. And designing, when you design something, you put things together. So, for example, as an electronics engineer, we use components that look similar to each other. And we even aggregate components together to deliver new products. We consider them new products when they have new functions that were not essentially performed by previous products, but the function needs to be very clearly distinct. So when you have, I don't know, an FM radio and you have now TV, one is, you hear voices from, one you look at the screen. So there is an obvious change. When we have a TV and we have a new version of a TV that has, I don't know, maybe more pixels or better color gradient or etc., this is not a new product, it's an enhancement for an existing product. So what am I hitting at? What I'm hitting at here is that it is normal in engineering, number one, that we use this similar components, and number two, that we typically aggregate components into modules. So for example, we have things that everybody has heard about, which are called ICs or integrated circuits. And what those are, they are chips. You can go to the store, you can buy chips, and no typical engineer will prototype a product by building it on the molecular level or on silicon. You will typically grab those modules and you will put them on a test bed and test it. And typically, even the final product will have a printed circuit board that will use plenty of those modules. Now, the experience in engineering is that you cannot at the end of the day say that the existence of similarities between different devices and the matter that devices come into versions that will cascade in complexity, and that even products like a radio and a TV can be, mapped into hierarchies that can look like nested hierarchies. And even if in the future we have machines that self-replicate, that the matter of similarity and hierarchical establishment of those things do not mean that they have evolved from each other or from common ancestors unless you establish that by independent evidence. Because the effects of design and the effects of ancestrally evolving into each other might be confusing. So the question will be whether the whole premise of evolution, which essentially primarily the Darwinian aspect of evolution, at least, there are other models like the Lamarckian or the endosymbiotic model, which are not Darwinian in nature. But at least the Darwinian model, which is the core or the centerpiece of the modern synthesis, depends on random mutations that are selected by natural selection. Random mutations are by definition random. There is no experience in engineering for something coming out from random mutations that are not guided by anybody. We have things in design called genetic processes or genetic algorithms, but they are guided by the programmer. So we don't have this model of completely random things, totally unguided by anything, producing something of specified functionality. This is not our experience with design. When we look at living organisms, when we look at living systems, we we sure find things that are extremely sophisticated with specified functionality. And this specificity of design means that there are different parts and that those parts interact in a specific way to deliver a specific function. Dan is talking about novelty. He says that evolution can explain novelty. I have so many quotations from pretty renowned evolutionary leadership, but I would like to quote the Royal Society meeting of 2016, where it was put on the table that the neo-Darwinian mechanism alone is deficient to explain all three categories that he has mentioned: phenotypic complexity, phenotypic novelty, and there is a problem with non-gradualism in the fossil record. So, no, I would say that evolution, which claims that from a last universal common ancestor to the modern human being, to Homo sapiens, we can explain this huge transition primarily by natural selection acting on random mutations to produce novelty and everything that we see around us, from complex things like eyes and ears falling under complexity, for completely new body plans and all the different kinds of mammals that we see around us, which is phenotypic novelty, or explain even the jumps that we see in the fossil records. So even the evolutionary leadership do not accept this as an established result of the Darwinian mechanism. And this is why other mechanisms have been proposed, in the neutral theory, self-organization, evo-devo, whatever, and a couple more. But I would claim that even those do not complete the picture. Yes, there is research, but it is unfair to claim today that the modern synthesis or even the extended modern synthesis can explain novelty. I will just give a fast, how much time do I have still, Dapper? Okay. I will just give a fast record of achievement or disachievement for the Darwinian model, which is essentially still, or the which is essentially still the core of the extended model evolutionary synthesis for 150 years. This is a theory that claims that it can explain everything from Luca to humans, and I have put them in few categories. We should expect that explains the transitions. However, there is no proper or complete explanation for almost all the major transitions, whether from protocells to the last universal common ancestor, from prokaryotes to eukaryotes, even from Luca to all the diversity, a huge diversity in prokaryotes, from eukaryotes to multicellular organisms, which Dan has commented upon. I will comment later in the back and fro. All the novel body plans and very sophisticated biological interactions like sexual reproduction, placenta reproduction, etc., that we see in mammals and other animals. We expect to see a smooth gradient of evolution without surprises, because these are random mutations that are typically small changes. We shouldn't expect massive changes, and we see many massive changes in the fossil record, the most obvious of which is the Cambrian, but not the only one. We expect innumerable transitions, even in the words of Darwin himself, but there are so many missing ancestor lineages that transitions are really rare if you consider how much we are looking for. We should expect that the genetic mechanisms allow for mutations to take place and allow for plenty of mutations to take place, at least to explain the huge explosions that we see in the fossil record. But we don't see this. We see genetic material having so many mechanisms to protect itself from damage and from change. Yes, some mutations escape, but not enough to explain what we see. We expect mutations to work, to produce something, but we see genetic mechanisms that will even mute or disable mutated genes. We expect an abundance of beneficial random mutations. We don't see an abundance of them. Yes, there are some examples, but not as expected. If everything will be at the end of the day explained by mutations, then we definitely are so far away from enough. I mean mutations that cause novelty, not just a change to a property of a protein or a few proteins. We expect plenty of junk genetic material, which was the predominant opinion. However, every day passes, it appears that it is a matter of our ignorance. And every day passes, functions for what has been previously branded as non-coding turns out to be, it doesn't need to be beneficial only by coding, but by regulation or any other function. And this is continuously happening every day. Which is essentially what I'm mentioning in number 8, that non-coding should be doing nothing, but essentially it every day points out to doing more and more. Efficiency should not be pronounced, because even if natural selection is a good search engine for a maximum or a minimum, and random mutations are generating the variety, we should expect that these systems arrive to localized minima or maximum of whatever function it's trying to maximize or minimize. But we find absolute and very good efficiency levels in living things that we even cannot match in our own engineering design. We expect at the human level gradual development of language that we do not see. We expect gradual development of intellect, but the next animal compared to our intellect is so far away. We expect gradual development of free will, like putting laws and regulations and rebelling against them and changing social cycles, etc. But we don't see this. These are features of humans. And this huge gap with humans is essentially a problem. Information theory teaches that when there are meaningful functional designs at a receiver, then there is a transmitter. So the question is, who is transmitting? And if we know already that the initial state of evolution, regardless what explains its arrival, which is the last universal common ancestor, we know for sure that it had much fewer genes than what we have in mammals. Then where did the rest come from? This is just a list of what's genetics and inheritance and epigenetics and breeding and drift and natural and artificial selection and adaptive responses are okay, but random mutation and natural selection is not. This is my presentation. Did I do well?
Well, you came in about 18 seconds under time, so I consider that a success, because I have to remain neutral, so I can't give opinions about how you did in the presentation, but I can say good job on not going over. All right, so
yeah, that's what I'm asking about. Yeah.
Well, at this point, everybody, we are going to go to an open discussion between Dan and Ahmed, which is going to last for about 32 minutes, and then we're going to have it'll probably actually end up being about 25 minutes or so of Q&A, and then we can have quick couple-minute final statements from our two debaters, and then that'll be it for this stream. But there is going to be a stream on my channel after this as an after show, to which both debaters will get links. I can't guarantee they'll both be here, but there will be also other people that I can bring on. We'll just chat about the stream, and my neutralness will go away on that stream. So if you want to hear my opinions, you have to go to that. Anyway, since Ahmed, you went last, I'm going to say that Dr. Dan, why don't you start us out for the first, whatever you have to say on the open discussion. And I'm not timing it yet, but if one of you starts to overtalk, I will start setting time limits.
Sounds good. So thank you for all that intro. Man, there's a lot of information in that intro. So there's a few things that I'm just going to leave on the table. Language, intellect, free will, that's like way beyond what I know anything about. So I'm not going to comment on any of that. I want to very briefly just make sure everyone's clear that Darwinism and even neo-Darwinism are extremely outdated at this point. So we mentioned the 2016 meeting, that the kind of takeaway there. I forget the exact wording of the quote, but it was neo-Darwinism is insufficient to explain whatever. I agree with that. Neo-Darwinism is insufficient to explain like all of biodiversity, because neo-Darwinism is from like the 1940s. So it just leaves out like a lot of stuff that we now know about how evolution works, right? So like the stuff that I talked about in my intro, a lot of that stuff like endosymbiosis, gene duplication, like a lot of that stuff is non-Darwinian and even non-neo-Darwinian. It's involves processes that are newer than that, related. Gradualism isn't like an expectation in evolution. Even actually, that goes back to Darwin. In one of the later editions of Origin of Species, he basically articulated the concept of punctuated equilibrium, even though he didn't use those words. That's what he described. So we know that gradualism isn't kind of a realistic expectation. There's a couple other small things we could talk about. We could talk about like junk DNA, which is very much a real thing. But I really want to comment on kind of the meat of the argument, which is, can we get new stuff? And I want to cite a paper from 2017 by, I believe the author, it's Rafik, I want to say Nimi, but I may be mispronouncing his name. This is from 2017. The title is Random Sequences Are an Abundant Source of Bioactive RNAs or Peptides. So what they did in this experiment is they basically randomly polymerized RNA and proteins, and I should say polypeptides to be precise, and they then tested them for any kind of biochemical activity, reactivity, whatever, and found a lot, right? So you may not get new things randomly via engineering, but in biology, you definitely get new things via, you could just randomly polymerize stuff, and those randomly generated molecules will have some kind of biochemical activity. We could also look at new genes across lots of different groups of organisms, because now that we know kind of what to look for, that we've kind of caught some groups in the act of getting new genes, we now know what to look for in terms of previously non-coding DNA becoming protein coding. So we can do this in plants. There's Rapid Evolution of Protein Diversity by de novo origination in Oriza. That's from 2019. We've got Origins of de novo genes in humans and chimpanzees from 2015. And hominid-specific de novo protein coding genes originating from long non-coding RNAs, that's from 2012. So we actually have like really robust evidence of all these new genes and all these different lineages. It's true that you do need to go from Luca to everything we have today, and that's last universal common ancestor, everybody. That's Luca, last universal common ancestor. Going from that to what we have today, you need a lot of new genes. Absolutely true. We have lots of mechanisms to get them, and we've documented those mechanisms. So I'll stop there and turn the floor back over.
All right, thank you. That was nice. So let's. How much time do I have?
I'm not timing this, but if it starts to feel like you've been going for a very long time, then I might stop you. But this is not a hard timed thing. But do try to be reasonably concise.
All right. So there is plenty that Dan has mentioned in his opening, and now he has put some more on the table. So I'll try to address a part of it in this one, and then maybe the next round I will address more if we close those ones. So let me first say this. This is like somebody bringing some electronic components, some resistors, some capacitors, some coils, throwing them on a table and saying, well, look, we have those things like, if you connect those things together, they will give you a circuit that can be a radio. If you do this to that, it will be this and that. Now, the question is not if. The question is, would they or not? So if somebody, for example, would polymerize a chain of nucleotides, RNA or DNA, and say, okay, some of this, if translated into proteins, or if we polymerize them into peptide chains, some of those proteins might as well have a good function. Maybe we produce this or that kind of protein, which exists in nature or not. This is not the question. The question is, living things have systems. So you don't need just a constellation of unrelated components. You need to see those components come together to deliver that specific function. And the ingredient for doing that is a designer. Now, the question now is, can natural selection acting on random mutations do the work of a designer or not? This is something that needs proof. It needs evidence. It needs very clear evidence, because it is against our experience that random things that are unguided by anything would produce anything. So this is one comment. The second comment is evolutionary paradigm or evolutionary theory, regardless what we name it, likes to use plenty of jargon that at the end of the day says nothing. I'm not attacking Dan. This is just a status quo. So, for example, Darwin very clearly said that if his theory is true, then there should be innumerable transitions. And he did say that having such gaps is a problem. And yes, somebody put on the table, even in the Royal Society meeting, that one of the 5 extra mechanisms is punctuated equilibrium. But what does punctuated equilibrium mean? There is no explanatory power in the word punctuated equilibrium itself. It just describes the fact that there are jumps in the fossil record and jumps in the gradual development. How can those jumps be explained by mutations and random and natural selection acting on them? There is no explanation. So just tagging a name to something, and this applies to punctuated equilibrium, to convergent evolution, to, I don't know, atavism, to so many terms that are used in the evolutionary literature, that all what they do is to explain the status quo. How come we have eyes that have developed in this creature and in that creature, and they have independently developed? Yes, it is convergent evolution. But this is not that I'm not asking about what is the name of the phenomenon. I'm asking about how, if just generating one eye for one creature in one lineage is so difficult for the Darwinian mechanism to produce, and then the explanation is convergent evolution, then you have now 12 or 50 lines of eye development, then you need to do 50-fold more explanatory literature for that. For each lineage, you need an explanation. How this one developed a compounded eye, how this one developed a simple eye, how that other one developed that kind of eye. Dan mentioned the non-coding proteins becoming coding proteins. And I'm not sure if I'm quoting him well, or non-coding DNA, I'm sorry, becoming coding DNA. And that long non-coding RNAs showing function. Again, one example of evolutionary literature using misnomers. So the literature first starts by saying that intergenic DNA is junk, and that whatever is produced from that is junk, and we have long non-coding RNAs, and then the literature says, well, guess what? Long non-coding RNAs do that function and this function, and there are small peptides that can be produced for them. And I've been just reading a paper that in one case that those small peptides would be generated in a specific way when a muscle is under stress to cause, for example, muscle regeneration. So the solution for that is not, wow, this is novelty. No, it's not novelty. We called something to be non-coding. We called it to be trash, or we called it to be junk, or we called it to be fossil DNA. And it appears that our understanding is wrong. There is no novelty. The function was there. And we are just learning more. Essentially, my point here is that evolutionary theory is in so many cases, especially when there are terms that have no real explanatory power, saying that what we did not have an explanation for, and now we have an explanation for, is evidence for something. Or sorry, what we did not know existed, and now we know existed, is an explanation. It's not an explanation for anything. It is something that we did not know, and now we know. Now give me the explanation, please. And the explanation is not evolution. So I will mention something that he specifically mentioned, because
we've now been going for quite a while, and I think this is starting to turn more into a rebuttal and a back and forth.
Sure.
So we're gonna try to keep it to one question or point at a time from here on.
That would be better.
So Dan, if you have one question or point, and then we can take it from there.
Yeah. So the big point I want to make there, I mean, so there's a couple little stuff like junk. We've known like non-coding DNA has functioned since the discovery of tRNA genes in like the 1950s. So like it's never been the case that we said non-coding equals junk. We know more functional stuff that's found in non-coding regions now compared to the 90s or the 70s or the 50s, but it's never been the case that non-coding equals junk DNA. But the big thing that I think I should mention here is the question you asked right at the top of for novel functions, it's not if they can form, but would they form under evolutionary conditions via evolutionary processes. And then right at the end, you circle perfectly back around to that of like, okay, you have the observation, but what's the mechanism? Well, the cool thing is we have the mechanism for this stuff. So we actually do have nuts and bolts mechanistic explanations for like why we have eyes in all these different lineages. And it has to do with, I mean, there's a bunch of different principles involved, but the short version is like for all these different types of eyes, you don't need 50 different sets of processes independently arriving at eyes, because we now know about this phenomenon called deep homology, where you have in different organisms, you have the same sets of genes underlying convergent traits. So there are a number of genes that are involved in eye development, whether you're talking about arthropods or chordates. It's the same genes. You could trace them back to the common ancestors. You can look, PAX6 is one of them. There's also the opsins, which are related to, they're basically signaling proteins. And way back in the early history of animals, these proteins experienced a mutation that allowed some of them, instead of being responsive to a chemical signal, they were became responsive to a photon. And now we have lots of diversification in different types of opsins, but everything that has eyes uses opsins as the actual photoreceptors.
All right, we gotta work on concision, everybody. So,
so
Dan,
my
could
point
you,
is,
could you
thank
name
Dapper.
some of the mechanisms that we have proposed?
Yes.
Okay.
Gene duplication, mutation, selection, random drift, and horizontal gene transfer across these different lineages. Many of these we have observed, and for the ones we haven't observed, we can make specific testable predictions, and the predictions end up being confirmed in terms of like, you could take a gene from this organism and put it in that one, and it does the same thing, even though it's one of them's a fly and one of them's a mouse, right? So you can make these predictions, and I think I know what the response to that's going to be, and we'll see if I'm right. So the idea is we know the mechanisms to get these novel features, and we can observe them and we can test them.
Okay, Ahmed.
Okay, thank you, Dan. So let me tell you this. So I really like what you said, because maybe with or without noticing it, you confirmed my concerns, and correct me if I'm wrong. So number one, the explanation for the existence of eyes cannot be deep homology, because deep homology is a word. Okay? Deep homology, you will need to tell me, for example, the first known eye in an animal, which is the trilobite eye in the Cambrian explosion. Show me its homologs in the ancestors of the trilobite. Now, the trilobite has a pretty complex eye, okay, and a magnificent eye, and we have it in the fossils. So we know that the trilobite has this eye. So if you invoke a term like deep homology, which is essentially plenty of homology, okay? So number one, you need to establish what are the ancestral forms of those eyes. If you invoke something like gene duplication, please tell me which genes have been duplicated. And then for my second argument, so the first one is using terms solves nothing. Show me the chemistry, or show me the mechanisms on the molecular level and on the phenotypic level, okay?
Well, remember, we're trying to keep it to 1 point or question per
Yeah,
go.
it's the same. I'm just responding to the eye, so I will respond to only what he has said about the eye.
I'm just saying you did literally say my second point, so that's why I became concerned.
Yeah. No, the second point just about the eye.
Okay.
So about, and then the second thing is that you said, okay, gene duplication. So the trilobite is the first thing that has the eye. So duplication of what? So we have putative creatures in the ediacaran, and we have some eye spots in, I don't know, algae or something. There is no complex eye for things. You mentioned one protein. What about the rest of the proteins? So let's not look at the table where we have some components that are scattered, because every electronic circuit will have a resistor. There are plenty of uses for resistors and capacitors. So if you find a capacitor in a TV and a capacitor in your washing machine, it has nothing to do with ancestry at all. It is just one component that's being used. The component here that we are looking for is design, and evolutionary theory says that design gradually evolves through ancestry. It is called common ancestry or common descent by modification via natural selection acting on random mutations. This is the central dogma. Now, if we invoke anything else beside this central dogma, fine, but show me how it acts. You invoke neutral theory, whatever, show me how it acts. The third thing you said about the eye is that we have signaling proteins and they have mutated in animals, etc. Well, show me how the first one has come in the first place. So signaling proteins, everything has signaling, but even the signaling proteins that are in the unicellular organisms, anything that senses needs something to pick up a signal. So saying that vision is using signaling proteins, okay, well and good, but the trilobite eye, beyond it has a specific kind of signaling, it really reacts to light, right? And it reacts to light in a way that forms a picture. And it has a complex eye that will provide this picture in a specific way that gives it an idea about what it sees around it in 3D. And then it has a neuron, and then there is a lens. It has a specific protein, it has crystalline, and in this lens will focus light. And the brain will understand it. There is a brain, okay, that understands it. So this is a system. What we are looking for here is not individual pieces of proteins. We're looking at
this one up, Ahmed.
the system. Yeah, I'm done.
Okay.
Okay, so now we're getting at kind of an irreducible complexity type argument here, right? It's not just that you need this component or that component. We I can give you the evolutionary pathway for some of the stuff you mentioned, like the opsins. It comes from these, they're called serpentine proteins, G protein coupled receptors. That's the ancestor thing. The kind of big idea with this deep homology is deep homology specifically refers to genetic homology that predates the morphological similarity. So it's the same underlying genes that predate the convergence in the morphology. And so when we say, show me the precursors to like the trilobite eye or something, the precursors are not to be found in the morphology. The precursors are genetic. And we can dig into the genomes of these different organisms, whether it's chordates or arthropods or even unicellular things, and even things that are different lineages like fungi. We can find the common ancestors to these proteins. You mentioned crystallins; those are related to heat shock proteins. So the way to get those is express them on the surface. So a lot of organisms have a surface layer that is contractile, right? It's the skin and the muscle together. Crystallins are found in muscles. Well, if you start doing photoreception, those crystallins are going to be in the neighborhood, and they end up with different functions involved in transmitting light. But without getting into kind of the weeds of this or that specific case, saying that this is now, it's a system, right, that we need this part, this part, this part, and this part. Now that's an irreducible complexity argument. And the problem with irreducible complexity is that we have documented the evolution of irreducibly complex systems. Like we have observed them in the lab, in human observable timeframes. The Lenski citrate example is irreducible. It meets Michael Behe's criteria for an irreducibly complex trait, but like we know how and when that trait evolved. We got to watch it happen in front of us in the lab and then go dig through the genetics. I'll leave that part there. And Dapper, if I could just say like one more sentence, two more sentences.
One sentence, but you can have up to 3 clauses.
Fair enough. So now I'm trying to, okay, how can I? All right. So the other kind of the thing that we're dancing around here is this idea of common ancestry versus common design. And the nice thing about that is a hypothesis, or those are two competing hypotheses that allow us to make testable predictions. Okay, that's a whole sentence. Sorry, I'm going to use a second sentence. So what we can predict with common ancestry is that we should have a nested hierarchy for both functional and non-functional components, right? Of whatever organism, whatever species, whatever lineage, we should see that nested hierarchy, whether it's functional or not, right? Because they share universal common ancestry. But with common design, we expect to see that hierarchy only for the functional parts. And for the parts that are non-functional,
this is like
or parts that don't have a functional significance, those should be uncorrelated across the different designed lineages. And what we find is nested hierarchies across the board rather than some correlated and some not. There's strong evidence in favor of universal common ancestry and against common design.
All right, I'm going to go now to a 3-minute time limit on these, so that's going to be strictly enforced, and we're only going to get like 2 or 3 rounds of that. So Ahmed, go ahead.
To be fair, I started. Well, I was going to say, to be fair, I started this round, so he should get one more with a looser time limit, and then we can tighten it up.
Okay, fine. Go ahead, Ahmed.
Okay, looks good. So once again, thank you, Dan, for the nice thing is that you're teaching this stuff, and I think, and hence you're putting forward to everybody what evolutionary theory has to say about those topics. So I have a nice presentation. I call it nice, maybe you don't, on my channel, which is about why contradictions or major issues with evolutionary theory. And one of the major things is contradiction. So let me point something out. So now we're looking at a complex system like the eye. I did not mention irreducible complexity, by the way, because I am criticizing the theory for using terms. I will not use terms. I'm just asking for this. We have a complex system, the vision system of animals. We know the first thing that we see in the fossil record is the trilobite. I'm asking a simple question. I'm not saying it's irreducibly complex or not. There are simpler eyes, so it's obviously not irreducibly complex. There are simpler eyes than the trilobite eye. So the question is, I'm not putting I can put irreducible complexity on the table, but I didn't. But the question is, we know that there is a complex system, which is the trilobite eye. We know that it is very early in the fossil record of animals because orthopods are essentially one of the earliest. And I am asking, did this system pop up into existence because the Darwinian mechanism does not allow for that? So your response to that, okay, maybe it did not pop up into existence phenotypically by magic. Maybe it was there, lurking in the genetic, composition of whatever was the predecessor of the trilobite, and then it came up onto the surface. So my response to that is that, sorry, this is completely against the basic premise of evolutionary paradigm. Because the evolutionary paradigm says this natural selection acts on reproductive advantage. And if genes are not complete, especially for a complicated system like vision, if they are not completely expressed on the lineage, so natural selection has nothing to select from. So there is nothing in the theory that says that the genes will just keep on putting the lens and the cavity of the eye and the photoreceptors, whatever they are. And then we have the neural link to the brain, and then the part of the brain that understands vision. And then the I don't know, neurovision, motion, coordination mechanism, because vision is too many things. There is nothing for natural selection to act upon. So if these things are happening undercover in the genetic material, they should, according to the theory, go away by exclusion because they will be by elimination, by extra mutations that will destroy them, or because they will be using too much energy in replication and they have no function. So this is my first thing. The second thing you said, okay, for example, we have muscles have... just about the eye. I'm replying to what he said about the eye only. So you said, okay, maybe crystalline, some muscles have crystalline on the surface of the skin. It is, again, the same thing I said. If you have a resistor on the table, so what? And number two, the crystalline on the muscles, does this answer the question? Are muscles using it for vision? No, it's not. Then you mentioned the Lenski experiment for the third time for irreducible complexity. I will need to comment on that because Lenski experiment, to me, completely debunks the evolutionary paradigm. The Lenski experiment, the popularized thing about it is that a set of E. coli, when deprived from glucose, they start metabolizing citrate, and this comes de novo to the stage, which is not the case because I have gone through the literature. What has happened is that the citrate machinery, the citrate metabolism machinery, was all over the cell.
Gonna finish the sentence. Yeah? Gonna finish the sentence, and that's it. We're gonna go to a 3-minute back and forth with one round of that.
Sure. So the citrate metabolism mechanisms were there in the cell. What we needed was the transport mechanism to bring the citrate from outside, and this happened because a transposable part of the DNA moved into a promoter part and triggered this action to promote citrate transport inside the cell. So
Okay.
there is no novelty. It was there and got activated.
All right.
So
Dan, you're going to have
yeah.
3 minutes, and then Ahmed's going to have 3 minutes, and that'll be the end of this section. Then we're going to go into Q&A. So Dan, you can start right now.
Thank you. That's a perfect example of what I wanted to kind of wrap this up with, which is kind of the big thing I want people to take away from this is that it's not one thing or the other in terms of like novel traits. It doesn't have to be a brand new gene or a brand new structure morphologically. What we see in evolutionary processes, by and large, is new emergent properties that are the result of different patterns and different arrangements of pre-existing components. So where do those components come from? I talked about that a little bit with de novo genes, with random sequences having biochemical activity. It's a whole field of where things come from originally. That's not my field. It's a lot of biochemistry, not my thing. But looking at going from Luca, last universal common ancestor, to everything we have today, a lot of what Ahmed just talked about is actually really well supported. These sets of processes, gene duplication, mutation, not selection, but not just selection. There's a lot of probabilistic properties involved where it's not so much that if it's not doing anything, it'll be selected out. It's just that it won't necessarily be selected for. So it just kind of like components in the genome or traits in the genome, sequences just kind of like kick around and they mutate and they're there and maybe they get transcribed and maybe not. And it's all kind of messy regulation. And all these things come together to generate lots of variation in terms of the traits you see in populations, right? That's the big idea. So you end up with these new emergent properties based on similar components. And over long time spans, billions and billions of years, you end up with, at the end of the line, genetic and morphological traits that are very, very different from the things they started with, billions of years ago at the beginning. And the thing I want people to take away, it's not one set of processes or one driving force behind this. It's many processes and many driving forces behind this all operating together. And all of those processes are quite well supported via hypothesis testing and experimental evidence.
Okay. Ahmed, you have up to 3 minutes starting right now. You may want to unmute.
Okay, if you can start it now, that will be great.
I restarted.
So, okay. Yeah. So once again, I'm going back to the same argument here, Dan. So just saying that those are many things coming together, number one. Not only one, but many processes is number two. There are emergent features, number three. And at the end of the line, you will find an I. This is not an explanation. This is a narration. Okay, we know that an I appeared. We know that somebody has killed this guy. It is not a matter of that he has been killed. It's a matter of who killed him. This is the issue. So that those processes are established empirically to prove their hypothesis. Sorry, of course, I do not agree. And I can take any specific example where proper novelty is claimed to have been generated by evolutionary mechanisms, and I will show you that it's not true. Through the evolutionary papers itself, and I have read plenty of papers. Okay, and then you see, you say pre-existing things come together, and those things that are not pre-existing are not explained by biochemistry. No, I disagree about that completely. Because yes, if you say that living things, I use the electronics example again, and we have a radio, an FM radio, and now it is using light bulbs, for example. And then a new component called a transistor comes into being. It didn't come by mutation of the light bulb. It's a completely new component. So let's look at this. There are pretty good evidence that there are actually DNA sequences, protein-producing sequences that have been proven to be protein-producing sequences in every living organism that do not have homologs at all. So they come completely de novo. Now tell me, where are they coming from? Because they are not pre-existing components. For example, in the human being, there is, if I recall correctly, more than 1,700 novel genes that are not available in its nearest, I don't know, common ancestor. It is specific to Homo sapiens. So even the claim that evolution just co-opts or whatever other term that you would like to use, pre-existing components, you have the same three problems. Number one, how does it do that? Number two, where does the design element come from? And number three, it is not even true that it's only pre-existing components. There is genes in the human, for example, and in many other animals that were not there altogether. Where is this coming from? I'm done.
All right, done with 4 seconds to spare. Very close. All right, so right now I'm going to open it up to questions, but first we are going to get to a couple questions that came in, one as a member chat on my channel and one as a super chat. So, I'm not exactly sure if this is an L or a capital I here, so I'm going to go with Pat L. Dennis, I think, or might be I'm not sure. Sorry about that, but has been a member for 6 months. I know that Ahmed is enamored with cytoskeletal motors. Viruses like Ebola, rabies, and herpesvirus utilize such motor proteins during infection. Were these functions designed, or did they evolve? Now I'm going to tell how we're going to do the Q&A. I'd like to do a 3-2-1. So Ahmed gets up to 3 minutes, then Dan gets up to 2, then Ahmed gets up to 1, and that's, whoever is the question is for. If there's a question where it's for 2 people, I'm just going to give everyone 2 minutes. And there you go. So Ahmed, you have up to 3 minutes starting right now.
Can you just repeat the question? You said it very fast. I didn't.
I'm sorry. The question comes. I'll skip the first section because it's not really part of the question. Viruses like Ebola, rabies, and herpesvirus utilize motor proteins during infection. Were these functions designed, or did they evolve?
Okay, I cannot confirm whether Ebola uses motor proteins or not. Now, the question, the whole thing I am saying is that let's now ask the question: where did motor proteins come from? Okay, and I'm not sure if you are even referring to the same thing when people are talking about motor proteins. This is a feature of eukaryotes, actually. So let's look at motor proteins, if this is your question. Motor proteins, you will not find kinesins and dyneins in prokaryotes, for example. They suddenly emerge in eukaryotes. And maybe Dan can tell me in his turn, where did those come from? But not only did they come in a huge amount of diversity. They come with cargo-specific adapters. They come with the microtubules that they walk on. They come with completely crazy integration with the ATP cycle to perform the walking action. They come with even functions that are related to the very existence of eukaryotes. They are very entrenched into the kinetochore cell division mechanism to the extent that you cannot have a living eukaryote that divides if you strip away microtubules and kinesins and dyneins and the kinetochore sensing mechanism. They are everywhere. They are necessary for neurons, etc. So now the question is, motor proteins used by whatever. If tomorrow they discover them in a kind of bacteria, I don't care. But today they are a eukaryotic feature. Where did they come from? This is the kind of novelty I would like Dan to explain to me. And the answer that maybe there was pro-A or pro-B precursor protein or very primitive cytoskeletal structures in prokaryotes explains nothing. It is like the radio and the bulb and the transistor and the TV. If there is a TV, you need to tell me where the design element came from, show me how the radio became a TV, and explain the mechanisms.
Okay. Well, now, Dan, you may have up to 2 minutes, and starting right now.
Help if I unmuted myself. So I don't have any specific comments on motor proteins or anything. I know of them. I remember learning about them in cell bio in grad school. But I don't have any specific comment on their evolution, except to say, I bet if we look in prokaryotes, we're going to find precursors. That tends to be the case for things that we think are eukaryotic specific. An example of that I can comment on is a eukaryotic feature called the spliceosome, which is very similar to something called self-splicing introns that we do find in some prokaryotic lineages. And we see this homology sequence homology in prior lineages without having the actual physical trait all over, the all different types of organisms, and human genes are a great example of this, where we have all those de novo genes in humans. We can find the homologous non-coding sequences in other primates, and they're protein coding in humans, and we can see what specific mutations led to that. I bet if we looked in prokaryotes, we'd find something similar for transport proteins.
Okay, now, if you would like, Ahmed, you can have up to 1 minute.
Yeah, thank you. The answer to this question, because I have looked deeply enough into motor proteins, is that the lineage of motor proteins stop at the last eukaryotic common ancestor. Yeah, there is no precursor to them that is functioning as a motor protein. You might find a part of this and that, but the literature that I have gone through says that this is a eukaryotic feature. So, and this is not the only eukaryotic feature. And the colossal failure of the evolutionary paradigm to explain eukaryotes at the end of the day caused the evolutionary community to accept what they have so many times rejected from Lynn Margolis, according to her endosymbiont hypothesis. But at the end of the day, it only barely explains mitochondria. The Golgi apparatus, the nucleus, the double membrane, no explanations. So once again, very little explanations, tip of the iceberg, explain nothing.
All right, 2 seconds to spare. I do want to say that I did get a question from Ahmed's chat from Got It, and we are going to be getting that as the first question that we get to after the super chatted questions. So our first super chatted question is from Raw Harley Quinn Energy for $10. And she says the 3 subfamilies of 6 genes are also conserved. 2 are involved in eye development and 1 in muscle development, both in mice and Drosophila. The common ancestor of both must have had them. So since this is not directed at a certain person, I'm going to have Dan go first because Ahmed just went last. You have up to 2 minutes. Go ahead and comment on that as much as you want for 2 minutes.
Basically, yes, this is a really nice example of deep homology, that we can see the same genes and the same gene families across extremely divergent groups of organisms. And I'll just note that we've actually done some experiments on things with like limb development, and specifically the one I know of a cool experiment with hand development. You can actually take genes from mice and Drosophila, and you can like swap them back and forth, and they'll do the same thing. So you can remove a gene from mice that basically causes them to not develop limbs, but you can replace that gene with a fruit fly equivalent, and it will kind of recover, at least in part, the lost function, which is super cool and evidence of the common ancestry of those different lineages, which again, we get the argument of common design, but then that's contradicted by the nested nature of non-functional regions. So it's common ancestry is the explanation.
1 minute. Oh, all right then. You only took 1 minute. All right, and Ahmed, you may have up to 2 minutes as well, starting right now.
Okay. So we have 2 creatures who share a common feature, and then we say, according to what Dan said, they must have come from the same common ancestor. We're saying that they must have come doesn't prove anything. Prove that they have come. Show me the common ancestor. The fact is that the phylogenetic trees that are claimed to be based on nested hierarchies that are proposed by evolutionary theory. Number one, which nested hierarchy are you talking about? Is it based on the full genome or cytochrome B or cytochrome C? And guess what? When you do it based on cytochrome B or cytochrome C, you get completely different results. And when you compare them to the one that is based on phylogenetic convergence, you get a completely different one too. So which hierarchy are we talking about exactly? So, for example, if you take a different criteria for human evolution, maybe you will not find, based on a different one, that humans are most related to chimps. Maybe they become most related to pigs, for example. It's the same thing. I'm not saying humans are more related to pigs, but I'm saying that the results will not have to be that you have a single uniform thing. And then the second thing that you said, that what makes nested hierarchies even better is that when you look at the coding or the non-coding regions, they seem to work. Well, guess what? Non-coding regions over and over again turn out to be coding regions. They have function. So the argument doesn't exist. This is a design that is based on efficiency. And at the end of the day, my prediction is 100 years from now, the human genome will have, and any other animal genome will not have anything that can be marked junk or not used. This is my personal expectation.
All right, with 14 seconds to spare, our next super chat question, which so far is our last one, so if we don't have any more before it's answered, we will be going over to our first question from Ahmed's audience. This is from Andreas Elv, I think. Sorry, not entirely sure if I'm pronouncing that correctly. This is for 50 secs, and he says, for Ahmed, in what way is a 51% complete eye not an advantage over a 49% eye in natural selection? And you have 3 minutes. Go ahead.
In what way? In what way is what?
Is a 51% complete eye not an advantage over a 49% eye in natural selection?
Of course, a 51% complete eye is an advantage, but I am telling you that the first eye that we see has no ancestor altogether. We have a trilobite eye, which is a very, very, very complex structure. It looks like an eye of a bee. Did you see an eye of a bee under the under a microscope, or under even those great things that you see on National Geographic or Discovery Channel? Look at the eye of the bee. This is how the trilobite eye looked like. And this is the first eye we have in the fossil record. Trilobites are one of the first animals that existed on Earth. And there is no ancestor at all. There is no eye before that. You have eye spots for algae. So it's, we're not talking about 51 and 49. We're looking at a complex eye, composite eye, and nothing. So this is the answer to your question. But if there is an eye that becomes a little better by a mutation, number one, show me the examples. And number two, I don't find a problem with that.
Okay.
There are examples for point mutations doing things, but very limited examples. Very, very limited. Not systems, not organs.
Okay. Now, Dan, if you would like, you may have up to 2 minutes. Nope. All right.
Let's
in that case, we're also moving. Not getting the 1 minute response then. So that means that we are now on to the first question that I have from the Finding Truth channel. This is from Got It, who says, question for both of you. Does genetics affect personality and criminal behavior? So we're going to start with Dan because Ahmed spoke last. So here you go, Dan. Good luck.
So this is kind of a nature versus nurture thing. The answer is basically not. I mean, like, there's personality is such a weird kind of amorphous thing. There's definitely behaviors that have a genetic component, more so in the further, like, not so much in not a lot in humans, but more so as you get into other organisms. You have like intrinsic behaviors that are genetically determined. And in terms of criminal behavior, like, no. There was a famous study of like, if you have an extra Y chromosome, like XYY, are you more predisposed to like criminal behavior? And no. Such a terrible study. It was a study of like a single prison in the UK, and it turns out XYY is super common. A bunch of people watching probably are XYY. You would never know unless you got genetically tested for it, because it has no discernible effects on anything. So yeah, no. For that one, no, I don't think so. It's all nature versus nurture. It's a whole lot of nurture.
All right. With 50 seconds to spare, that's good. And Ahmed, if you would like, you can have up to 2 minutes to also talk about this question. Go ahead.
Very interesting question. So now the question is, I'll a little bit broaden the question. Where does behavior come from altogether? So evolution claims that it explains living things, right? It explains the origins of species. It explains how we have monkeys and beavers and mice and elephants and bees. Now, I have an argument from beavers, and this is the nickname I call it. I even had a discussion about that. And look at everything, a single animal that you find around you. Why does a cat get born as a cat? Why does it run after mice? How come a beaver knows how to build a beaver dam and completely terraform the forest? How would a bee understand sign language? Why does a salmon fish know that it needs to migrate and leaves the ocean and goes upriver and against the current of a waterfall to lay eggs in a river that
1 minute
and then dies, etc.? All of those behaviors, thanks to Dan for saying they are not genetic, because if they're not genetic, where are they coming from? naturalism and methodological naturalism would like to attribute everything to a material object. If information of living things are coded in genetics, and this is not genetic, from where is any behavior coming from? And those are behaviors that come with those creatures upon arrival. They do not learn them. So this is a big question for evolutionary theory.
Okay. Our next question is from the Dapper Dino chat. This is from David DB, and then there's a lot of numbers, so I'm just going to say numbers. Who says, for Ahmed, are the apparent nested hierarchies in design things not a result of designers learning new things and getting better at their jobs? If so, how does this work for God?
Excellent question. So, yes, the nested hierarchies for human designers show that human designers have a continuous process of innovation and a continuous process of enhancement for what they have innovated before, and hence they would apply this to existing designs such that those existing designs will be you can build a nested hierarchy for them. How does this apply for God? It doesn't. Why? Because humans, when they do those changes, number one, they create things that they need. Okay, so when their needs change, they create things that and number two, they enhance their designs. But even when you look at the final reckoning of evolutionary theory, you will find when you tell them that humans are better than, I don't know, mice, they will say, no, there is no better or not better. There are mice who are at the edge or the verge of their evolutionary path, and they will get better, and humans who will essentially get better, and you will look at transhumanism one way or the other. Prove it. Prove it. No, what we see on the ground is that a bacteria is extremely versatile, and that a monkey is extremely versatile, and that a human is extremely versatile. So obviously God creates many forms, and each form is a piece of art of perfection. This is what it is. Perfection within the limits of this world being a world of death and decay. He has not claimed that this is paradise. So animals have to die, and there is disease, and there is mortality. But within this framework, you have utmost beauty and utmost perfection. Thank you.
All right. With over a minute to spare, if you would like, Dan, you may have up to 2 minutes. Go ahead.
Yes, this one I'm thrilled to comment on this one because this gets at the common design versus common ancestry thing. And again, we have a testable hypothesis here because if you have common design, right, you have separate things that don't share common ancestry. And then within that, you're going to have some components that are constrained by their function, right? They're going to stay the same because they're designed to do a thing, and they're going to maintain doing that thing. But you have other parts that I think we could agree that you can change the sequence without losing the function, is how I'll say it. Because Ahmed earlier said that, well, probably 100% of the genome is functional. I don't think that's correct. I think it's 10%. But I'll give you that framing and say there are sites like synonymous sites with encoding sequences where we know that selection for the bases at those sites is extremely weak. So you can have an A, you can have a G. It doesn't really matter. It's not going to be selected for or against one way or the other. Those sites should vary in a correlated way within these designs' lineages, right? Because they're going to share a common ancestor and a common history of mutations. But across the different design lineages, those sites should be uncorrelated with each other. What we see instead is that the nested hierarchy that we can generate using genes or regulatory sequences, right? Something we agree is functional. You could take those same regions that are not constrained, and you could generate that same nested hierarchy across groups that should not, according to creation, share common ancestry. So we have a specific testable prediction from a common design hypothesis that is wrong, right? That prediction is incorrect, and the prediction from common ancestry is actually what we see.
Okay, 2 seconds to spare. If you would like, Ahmed, you may have up to 1 minute.
Yeah, so thank you for the comments. Essentially, there is nothing in the paradigm of creation that says that any specific two categories of creatures have to or have not to have a common ancestor. But there is a specific assertion that everything is designed by the same designer. This includes viruses, bacteria, eukaryotes, multicellular organisms, chimps, humans, mice, arthropods, everything. So it is well expected, according to this hypothesis, or according to this theory, that all of them will have the fingerprint of the common designer. And just to wrap it up, it occurs that, as funny as it seems, that the same genetic material, DNA, RNA, information-bearing molecules are used.
We're almost certainly not going to be able to get to all of the questions that have been asked. We have one more super chat question, and we might be able to get one more after that, depending on how long it takes. So, yeah, I'm sorry for everyone who asked questions that aren't going to get answered, but I mean, what can I say? Some people did pay for their questions, so yes, they're going to get those answered. Our currently last super chat is from Vandalia 1998 for $5, who says, Finding Truth said if you compare different things, we get different nested groups. So why do we group genetics and morphology and others the way that we do? And you have up to 3 minutes. Go ahead.
I think you should ask this question to Dan, because it's well known that if you use, for example, cytochrome B or C, you will produce different phylogenetic trees. And if you use morphology, you will generate a different one. Sometimes the differences will be obviously not huge differences, because essentially phenotypes are expressions of genotypes. So you expect them to be not so far away, but there are differences. And if the hypothesis is true, then those differences should not exist. And it doesn't get any better with actually, this debunks many of the arguments of the evolutionary paradigm. So I will leave it to Dan to further comment on that.
All right, Dan, you have up to 2 minutes. Go ahead.
So first, it is true that different parts of the genome will get you different hierarchies. Sometimes. Most of them end up 99% the same, but then sometimes the relationship over here or over there is different. And we know why that's the case. So you have, for example, a mechanism, a process called incomplete lineage sorting, which we can model mathematically and then see that the pattern that we see in nature actually matches what we would expect if it works the way we think it works. We also have horizontal gene transfer, right? You have a lineage over here. If you've got a lineage over here, well, a gene from this lineage gets into this one. That's going to lead to, if you follow that genetic pathway, that's going to lead to a different hierarchy, a different phylogeny, right? So not only do we have theoretical explanations for why we see differences for different parts of the genome, we have mechanistic explanations for that have actually been experimentally verified.
Okay, and Ahmed, if you'd like, you can have up to a minute, and then we're going to go to our closing statements, which are going to be 2 minutes each. So go ahead.
Yeah, I would like to do this comment also about, Dan said about the junk DNA and maybe not the whole genome is functional. I like this thing, and it also relates to this question. Let me tell you this. For example, myself and you, Dapper, have talked briefly previously about endosymbiosis, and we were talking about, this poor bacteria that became mitochondria and lost the majority of its DNA because it became not functional, because now the eukaryotic nucleus is producing its DNA. And your argument, for example, at that time, because when I objected, you said, well, the DNA was no more needed, so it was lost by time. So my question is, if we have 90 or 10 even % of the DNA of any creature that's completely non-functional, why should it exist in the first place? According to the very premises of evolutionary theory, it should go away, and it doesn't. So since it's not going away, it is functional, and the whole matter of hierarchies is not proper proof for evolution.
And time. All right, so we are going to set up 2 minutes we're going to have Dan go first, because that has been the tradition. So Dan, you have 2 minutes to give your closing thoughts, and then Ahmed will give his closing thoughts. So go ahead.
All right. So big picture stuff. The topic that we talked about today was the evolution of novel features, and could that happen, and how could that happen, and what evidence do we have for that happening? The big thing that I want viewers to take away from this is that, well, first, we have directly observed the evolution of not one new feature, not a few new features, but like lots of novelty. in different groups of organisms, biochemical traits, genetic traits, morphological traits, physiological traits, and not just in microbes like viruses or bacteria, but also in things as complex as animals, right? We've observed a lot of, like, directly observed the evolution of a lot of new traits. Now, that by itself doesn't tell you anything unless we understand the processes underlying it. And the case is that we do understand the processes underlying it. We not only have evolutionary theory, kind of, capital E, capital T, evolutionary theory that proposes the processes and mechanisms that are underlying these observations, but we've actually directly tested and observed those different mechanisms. So we could dig into the genomes and see where gene duplication, for example, explains a specific trait. We can test predictions based on universal common ancestry or deep homology and show that those proposed mechanisms are actually doing a good job explaining what we see. So it's not just, can novelty evolve? Yes, we can watch that happen, but how? Via a number of different evolutionary processes that we've talked about today, for which we have direct observed evidence. Thank you.
All right, 18 seconds under. And now it is time for our final statement from Ahmed before we get out of here. Oh, you're muted.
Sorry, if you can start the timer now.
Yep.
Yeah, okay. So when we talk about novelty, we are looking at a journey that starts from Luca, which has been estimated to have some 300 plus genes. I don't remember, 335, 350. To human beings who have like 20, 25,000 genes, and those are the ones that we have discovered. We are looking at single unicellular organisms that have minimum functions of everything, to the human being that has all this amazing intellect, language, dexterity, beauty, and against all the odds rules the planet. He's not the strongest, he's not the fastest, he's not the sharpest vision, he's not the sharpest anything, but he has those unbelievable capabilities that makes it through the planet and change the planet and maybe invade space. When we're talking about novelty, we're talking about this. Can this be explained by evolutionary mechanisms? No, it cannot be. And it cannot be for every example that Dan has mentioned, whether because the example does not qualify as novelty or that the example is not through an evolutionary mechanism altogether. It is just something that we did not know, and now we know. The fact is, the main explanation that evolutionary theory puts on the table is that mere chance, with sheer luck, has produced eventually the human being from something that looked like a bacteria. This is a very large claim. It needs very, very solid evidence, and the extended modern evolutionary synthesis paradigm provides nothing close to even starting to explain this at any of the junctions, the important forks that I have explained in my presentation.
All right, just exactly on time. Well, thank you very much to both debaters. Thank you to both audiences. I know that there were a lot of questions that I couldn't get to, and I do apologize, but we do have a hard time limit on this, which we are right up against. So I just want to say there will be an after show on my channel. It'll probably start in about an hour, and it will... If you are on my channel, it will direct you to that if you want to click on that and get ready for it. Both of these speakers will be invited. I can't guarantee whether or not they'll be able to show up because they do both have responsibilities and, their own things to do. So we'll do what we can. But if you want to continue this conversation later, it will be on my channel in an hour. Thank you very much. And if either one of you have any last announcements for your channel or anything, go for it. I'm done.
Yeah, I would rather that Dan goes first.
Thank you. No, nothing. I don't have any. I got a couple things kind of behind the scenes that I'm working on, but nothing specific coming up. So, just thank you, Dapper and Ahmed, for a fun conversation.
All right. So, first, thanks to Dan and thanks to Dapper for moderating this. Dan, thank you for a reasonable conversation and a nice discussion of what the theory says and what it does not say. And what I'm working on now, I'm proceeding with my evolution course, which we are at session 5 out of probably 20. I issue a session every couple of weeks and then have a live stream on Saturday to explain it. And typically, I also bring renowned scientists. I had Dr. James Tour, for example. I had Dr. Fazrana discussing specific things and relating them to the course. And apart from what I'm also doing on the channel about things related to religion and philosophy, I have Dr. William Lynn Craig, who came once and is coming again on 3rd of November. And this is the first time I announce it, discussing divine aseity, which is why God is independent of anything. So plenty of other scientists also coming to the channel in the near future. And once again, thanks to Dan and Dapper. One small comment, Dapper was related, referring to super chats. So for people on my channel, this is because his channel has super chats, which is the ability of people to ask questions and donate money. I don't have this on my channel, so the super chats relate to his, and because my channel is not monetized at all. But thanks, Dapper, for a fun conversation, and maybe we should discuss endosymbiosis at one time. Dan, definitely I would like to have another live stream with you. We have opened so many things and so many loose ends. And you're a fun person to talk to and pleasant to discuss with.
I just want to take a second to thank my channel members and patrons, especially those pledging $20 and above. Bob Knob, Bent Hovind, Ian Chen, Chris Love, Sphincter of Doom, The Venerable Bede, and Res Instance. My channel members and patrons help make this channel possible, and without their support, this channel really wouldn't exist. If you would like to help support the channel as a member or patron, there are links in the description to both join the channel as well as join the Patreon. Patrons and members get mentioned in these credit scenes, as well as getting early access to my scripted videos, and if you pledge $10 or above, you can also get access to various 3D assets that I create for Blender, both for use in the channel, as well as just general giveaways to my supporters to help in any Blender projects they might have. If a monthly or annual pledge isn't for you, then there is also a merch store linked to the description. And if none of that's right for you, please just like and subscribe, because every like and subscription really helps the video out. Thank you.